This video is about immunity in health and diseases, immune response against viral infections, and this particular study note is developed by MHRD, APG Panchala team, Government of India. So I would like to express my sincere gratitude to the development team for developing this study note. Thank you so much. Viruses and immunity related to viruses is very important because we need to find vaccine against some important viral diseases like HIV, which is not present, like vaccine is not present. So that area is open for research. So I would like to explore this topic a bit more. In our day-to-day process, we are exposed to large numbers of virus which has the capacity to infect and cause cancer. Viral invasion usually occurs at mucosal surface lining, the respiratory, gastrointestinal, and genitourinary tract. Primary infection can establish into disease in absence of adaptive immunity to viruses. The response of immune system to virus is usually divided into innate immune response and adaptive immune response based on type of system activated. The innate immune response provides a generalized form of host response to various infectious agents. The innate immune mechanism acts to control the spread of infection and downregulate the viral replication. The recognition of virus or viral particles in innate immune response occurs via certain strategies based on pattern recognition receptors (PRRs). These receptors identify conserved microbial structures found throughout the same classes of microorganisms. Some metabolic products play a crucial role as a part of PRRs, and they are characteristic of particular microbial class. In case of viruses, the various viral molecules are produced in the infected cell. However, the main targets for recognition by PRRs are viral nucleic acids. The innate immune system detects the viral RNA also. Viral nucleic acid is found in the endolysosome and cytoplasm of the host cell. On the other hand, host DNA is not found in these places. In cytoplasm, sensing mechanism for both RNA and DNA of virus are present. The sensing mechanism can trigger immune response in the cytoplasm. In addition, the change in cellular processes, for example, acute storage of host protein synthesis, ER stress, or change in activity of ion channels also serve as sensing mechanism. Once the virus is recognized by the innate immune system, it leads to two-step elimination method. First, it leads to induction of antiviral response that is mostly mediated by type 1 interferon. Secondly, it can cause activation of adaptive immune response that would result in long-lasting antiviral immunity and also antigen-specific immunity. The priority of host response to viral infection is to eliminate the infected cells. The major response affected is the cell intrinsic mechanism based on type 1. However, this response can also be achieved in infected cell via cytotoxic lymphocyte, NK cells, and CD8 plus cells. Viruses are also prevented into host cell via neutralizing antibodies. Monitoring of viral infections. In mammals, multiple mechanisms exist for identification of virus. PRRs are one of the common mechanisms that are mostly utilized to detect the pattern associated with viral nucleic acid. Innate immune method of viral recognition includes both cell intrinsic and cell extrinsic based on type of cell involved, infected or not infected. Inside the cell, sensing mechanism include node-like receptors, NLRs, RIG1-like receptors. These receptors are generally activated in infected cell. TLR, toll-like receptors, and C-type lectins, transmembrane receptors, are involved in cell extrinsic recognitions and that are mediated by macrophage, dendritic cell, and plasmacytoid dendritic cell. You can see this representation which indicate various receptors involved in monitoring of viral infection and self-recognition of virus. RNA virus infecting a cell is recognized by myelin differentiation associated gene 5 MDA5, retinoic acid inducible gene 1 RIG1, and cytoplasmic RNA helicase. The receptors belonging to RIG1 like receptor are expressed in large number of cell type and recognize viral RNA helicase domains. RIG1 also possesses a receptor domain at its C terminus responsible for its inhibition, and it also contains a catalytic domain to initiate antiviral signaling. Mitochondrial antiviral signaling protein is a membrane protein that acts as an adapter for MDA5 and RIG1, which are distributed on peroxidases and microbial membranes. FADD and caspase 8 to 10 are protein kinase that acts as targets for MAVS. Cytoplasmic viral DNA sensors. There are multiple DNA sensing mechanisms for DNA from various microorganisms like bacteria, virus, apoptotic host cells, and synthetic B form DNAs. Example: poly-DA-DT. B form of DNA triggers type 1 IFN through NKKB and IRF3. DAI, commonly known as DLM1 ZBR1, is a molecule involved in intracellular DNA sensing. Then cyclophilin A, structure of viral infection. In some cases, viral protein serves as structure of viral infection. Cyclophilin A is a peptide phosphoryl isomerase that catalyzes distance isomerization of proline epitopes on target proteins. In dendritic cells, the innate response to HIV-1 depends upon the encounter of newly synthesized HIV-1 capsids and cyclophilin A present in cell, leading to type 1 IFN response. However, this capability of dendritic cell is lacking in CD4 plus cells. Therefore, this mechanism is not utilized in human for type 1 IFN production because human DCs are resistant HIV-1 infection. Cyclophilin A is incorporated into HIV-1 virion by binding it to capsid protein of HIV-1 to utilize it for its infectantly, because virus lacking this would be defective in the reverse transcription of viral DNA. Virus recognition outside the cell. Role of TLR. Toll-like receptors are employed as pattern recognition receptor for majority of microorganisms against the evolutionary preserved molecular pattern. The number of TLR present on a cell varies. In mice, there are 11 functional TLRs, and in case man, there are 10 functional TLRs. All TLRs are basically type 1 transmembrane protein and an external domain that contain leucine-rich repeat that recognizes pathogen-associated molecular pattern. In a TLR, the transmembrane domain guides its localization, and cytosolic domain contain a toll interleukin 1 receptor domain. This domain leads to activation of adaptive protein and produce pro-inflammatory cytokines and antiviral cytokines. Type 1 IFN production lead to activation of genes that suppress viral replication. TLR 1, 2, 3, 4, 5, 6, 11, and 12 are expressed on cell surface, and TLR 3, 7, 8, and 9 expressed in endolysosomes. TLRs in some viral invasion are utilized as a passage for virus entry rather than to be acting as TAMP's receptor. Many viruses are recognized by TLRs that are present in endosomal membrane that comes in contact with the nucleic acid of viruses after their endocytosis in the form of virion. Role of C-type lectins. Plasma membrane, in addition to DLR, also contain another type of receptors that is capable of bonding to viral antigen through their carbohydrate recognition domain and initiate CA2 plus dependent ligand binding. These receptors are C-type lecithin (CLR). There are various forms of CLR whose reactivity varies from each other. For example, some CLRs do not bind to CA2 plus, and some of them are used for inducing signal, while others prefer to cause endocytosis of pathogens. CLR responsible for endocytosis include mannose receptor and Langerhans both of which are expressed in CD8 plus DC and Langerhans cells respectively. HIV viruses are cleared by binding to Langerhans a Langerin molecule present on Langerhans cells. Signal-inducing CLR includes DC sign and lectin-1. DC sign bind to Ebola virus and HIV-1. DC sign does not cause activation of NFKB or cytokine production but modulates these activities through other PRRs. Role of cytokine in an antiviral response. Role of tumor necrosis factor. Tumor necrosis factor receptors have cysteine-rich extracellular binding domain. The binding of TNF to TNFR receptor results in formation of a trimer. The death domain is present in the TFR1 cytoplasmic site that recruits an adapter molecule called TRADD, tumor necrosis factor receptor type 1 associated death domain protein. TRADD is responsible for the assembly of two separate signaling complexes receptor interacting protein or FAS associated protein with the death domain pro-caspase 8 and TRAF2. TRAF2 activates JAK signaling pathway and RIP1 activate NKKB. Role of type 1 IFN. An appropriate immune reaction towards virus on vertebrates require the secretion of virus in vertebrates require the secretion of interferon type 1. IFN is responsible for inducing antiviral state in infected and uninfected cell lysis in proximity. It also acts as a mediator of adapter immune response. The other type of interferon type 2 and type 1, 2 and 3 also act as potent antiviral cytokines. Role of inflammatory cytokines. There are good number of cytokines that are involved in inflammatory response that ultimately result in production of antiviral effector molecules and simultaneously stimulate cellular recruitment and phagocytosis of infected cells. In addition, it also leads to adaptive immune response by activating cytotoxic T lymphocytes and neutralizing antibodies. Inflammatory cytokines IL-6, IL-1 beta, and TNF that are secreted by infected DC and macrophages. These cytokines are responsible for causing fever due to release of prostaglandin E2 acting on the hypothalamus that cause increased heat production from brown fat and vasoconstriction to prevent heat loss. Fever prevents the replication of virus at higher temperature. Role of Tetherine and Vipidin. Tetherine undergoes high expression on stimulation by IFN. It inhibits the release of a retrovirus particle in the absence of viral protein unique VPU. Tetherine binds the lipid and coordinates the change to inhibit the release of retrovirus particle. VPU uses the beta TRCPE3 ubiquitin lipase complex and induces endosome trafficking, which is responsible for removing the tetherine from cell surface, disabling it to restrict the release of enveloped virus. Tetherine limits the release of retrovirus fibroheparin virus inhibitory protein. Endoplasmic reticle associated interferon inducible is secreted in response to IFN and is responsible for inhibiting replication of viruses like HIV, dengue, alpha virus, influenza virus, and hepatitis virus. Heparin suppresses activity of farnesyl diphosphate synthetase and thus disrupt the release of influenza virus by disorganization of immune lipid rafts. Role of APO-BEC family in innate immune. APO-BDC. Appropriate apolipoprotein BMRNA editing catalytic polypeptide is a form of cysteine deaminase that has one or two catalytic deaminase domains. The expression of ApoPEC increase on stimulation by type 1 and 2 IFN and has a constitutive expression on various cell types. Role of RNA interference. One of the prominent antiviral defense mechanism in some plants, fungi, insects, and nematodes is RNA interference. RNA operate in two steps. The first step results in identification of viral dsRNA by molecule known as dicer endonuclease family that cleaves dsRNA into siRNA. Second step results in the incorporation of siRNA into RNA-induced silencing complex RI RISC. This complex guides the RNase enzyme AGO to a complementary sequence in viral RNA for its degradation and cleavage. There is no involvement of RNA found in mammalian cells infected with virus. A mammalian cell also lacks RD prepinase amplification of siRNA. Then adaptive immunity to viral infection will be there. Insight to adaptive immunity to virus infection, B cell response to viral antigens, recognition of viral antigen by T cells, viral antigen neutralization by antibodies, B cells and primary viral infection, B cell and secondary viral infection, when how viruses evade immune system, change in antigen displayed on surface, ability to complement inhibition, cytokine inhibiting proteins and degradation of proteins, MHC class 1 downregulation, MHC class 1 molecular retention in cell.